Hey, good morning, everyone. Thank you for joining. And like I said, today we will be seeing about how to prepare a species identification report. Okay, as a Barcode Bioinformatics division, this is something important department that connects the wet lab with the clients. Okay, so it is here in Bioinformatics division. Actually, the species identification report and gene identification reports are prepared. Okay, so far in our summer internship, we have seen how to work with the machine output file, ABI files, how to manually inspect the chromatogram and edit and manipulate the sequences skillfully, how to identify and call the appropriate nucleotides using chromaslide, and how to merge. The yesterday's bioedit is all about how to merge both forward and reverse sequences and to one. And finally, we have after merging, we need to know how to take it to the blast analysis and how to find the species name. After finding the species name, we have seen how to extract the sequences retrieved from the blast results, how to prepare the FASTA file by introducing, underscore between spaces, how to prepare a phylogram, and how to prepare a pairwise data. And today we will see how to prepare a species identification report and what are the files that needs to be uh, mailed to the clients. Okay, so imagine a client who is working with a bacteria. He wants to identify his species. So the client will send you the samples in plate or tube. So wet lab department, they will assign a code. Maybe say, for example, if the client's sample name is S1, wet lab department have a COML codes. They assign a code for S1 and they will take it for DNA isolation, purification. They will use the DNA template. If it is a bacteria, they will go for 6NS rRNA primers. They will amplify in the PCR. They will purify the PCR. They will split the sample into sample amplicons into two, like we saw yesterday. Forward single standard amplification for PCR. They subject every sample as two reactions for Sanger sequencing. Once sequencing is done, you will have a forward primer, forward sequences and reverse sequences. So you have to manually inspect the chromatogram, edit it using BioEdit. Usually they may, sequencing department may do this, or sometimes they will send the machine output file to bioinformatics department. Okay. So here, if you are working as a bioinformatician, if you are, working for Barcode Biotech or any biotechnology company involved in DNA sequencing, they may send you the machine output file or a notepad file as an email, as you have seen here. I have got an email, actually it's an actual client file. Yesterday I have shared it with you. So I have downloaded this, I have opened this, I have a forward and reverse primer. I will quickly recall what are the things we have to do, and finally I will tell you how to make the species identification report. Okay. So I have downloaded the sequence, and like I said yesterday, copy the reverse sequence, bioedit, import into bioedit, reverse complement it, copy the complement, and paste it in the notepad. So notepad will have a forward sequence, reverse sequence, and the reverse complement. Delete the reverse sequence. So you will have a forward sequence and reverse complement. In the Word file, the last 10 fragment or 12 fragment of the nucleotides of the forward, go for Control F, find it. It will be here. From here, leave till AAG, and from here, GTT. I have trimmed it, delete this. Now merge the sequence. Okay, that's how the forward and reverse are merged. After merging, we are taking to the blast analysis. I got 100% query coverage and 100%, nearly 100% similarity for leuconostoc mesenterites. So note it down. This is leuconostoc mesenterites. For microbiology people, mostly, they will be interested in discovering new species. When do you know your bacterial species is new or not? When the similarity is less than 97%. Okay. If you find this sequence is sharing 97% similarity with the leuconostoc mesenterites, meaning that bacteria can be leuconostoc, but they are not mesenterized for sure, because species cutoff is 3%. 97% is actually 3% variation, so they cannot be as same species. And also, uh, whatever species it can be in the leuconostoc, but it is not available in GenBank. That's why it is the maximum identity is 97%. So this is the first cue you should know that you may be working with a new species or novel species. We have other assays like DNA high precision, whole genome sequencing to verify new species. Then you can propose your own species name, okay, because it's a new species. That's how discovery of new species is done. And BLAST is a first window that give you a glance of whether your strain is new or not. So note down the species name. Then I would say go for another BLAST. Now selecting, now you know it is a 6' rRNA. Till now you do not know what it is, right? So now you know it is 6' rRNA. Go for select 6' rRNA database and go for one more BLAST, because see, GenBank may have 6 gluconostoc mesenterite species, and these are other gene sequences from gluconostoc. Okay, so when you have see, so only the type species are displayed here. Now you can select if a maximum of 5 sequences from same species and 5 sequences from same genera of different species. So that's how I did the selection here. Okay, if you scroll down, you can see I have selected other species in gluconostoc. Then we'll go for download and download the sequence. Now after downloading, open the sequence, put your own sequence, the query sequence you used from the sequencing department, which I have marked it as my sequence here. Then all the reference sequences. I have incorporated underscore symbol here to prepare the FASTA file. Once the file is done, go for ClusterLex analysis. Okay. So you guys know now the output of ClusterLex file is ALN file, which has to be fed into Mega for other analysis. Okay, other alignment adjustment. Yesterday, one of the intern actually shared this file. You see here, this is not Luconostock file. This is really different file. But you see here, my sequence, the query sequence I have is somewhat weird. Okay, even though they all belongs to same species, how come they have a gaps here and there is no continuous star mark? Okay, see, many people who are submitting sequences and publishing sequences in GenBank, they either submit forward strand or reverse strand. Okay, the chromatogram I have sent you, and many of you have successfully identified chromatogram C is a usable one, and the chromatogram A and B are not usable one. So congrats for that. Chromatogram C is actually a reverse file. All my reference sequences are forward files. So that's why when we introduce this reverse sequence here, it's not aligning. So how to address this issue? Yes, this issue should be addressed before proceeding with the multiple sequence alignment. So you select this sequence. You see here in Mega, you have reverse complement option like that in BioEdit. Okay, you just right click this and go for reverse complement. And once you click reverse complement, see the alignment now. After reverse complementing, now I have converted the reverse sequence into forward sequence by reverse complementing, and now it is perfect. So this has to be done. Okay, just keep in mind. So I'm opening Mega after cluster alignment. I'm importing my mesenterized, Luconostoc mesenterized sequence, do the alignment, remove the gaps, and then we import the file to phylogenetic analysis. In the phylogenetic analysis, you guys know where to select NJ tree method. And once the tree is done, make sure you complete the species name. Many times after 35 characters, they may not take the remaining characters. You have to manually edit by double clicking and make sure every species is complete. And you also see here this branch length here. Okay, this is not sufficiently seen. So what I will do, I will go here, layout, try to increase the layout for better resolution. See here now and see here now. I have just moved this tree width to 1 unit. Okay. Usually tree height, never increase it. Okay. Try to fit the entire tree in one page. So if you want, you can reduce the tree height. If you are increasing the tree height, it may go to 2 pages, or the tree will be little not elegant. So just increase the tree width. And there is another thing a client might be expecting from our side. After drawing this, go scroll down the left side menu and display caption, where you will see something like this. You can copy this and paste it in the notepad file or a separate Word document. Okay. So what is this tree caption? It will tell us how many nucleotides are involved. They have used NJ method. The pairwise distance data is calculated by Chimera2 parameter. How many data set positions was available after deleting the gaps. Every information will be there with the references. So client, when you're sending a tree file of a publishable quality, they may expect us to send the captions because they need to incorporate in the manuscript. Okay. So once tree file is ready, see, I told you in the beginning of this presentation, the client sample is S1. The wet lab department actually decodes it. Actually, the reception department will decode and give to the wet lab. So when you are making a phylogenetic tree, better you use the same code. If the client has given the sample as S1, better put S1 here. See, this tree is a pakka proof that my sample is a leuconostoc mesenterites, because proof number one, it shared 100% similarity or nearly 100% similarity during blast analysis. Proof number two, it is sitting within the branch. An ideal phylogram should be like this. It is sitting within the branch of leuconostoc mesenterites, which means it is leuconostoc mesenterites. If I introduce the same sequences of leuconostoc, see gelidium here at the bottom of the tree, two gelidium, same species are cladding in same branch. This is a proof. And hence, in the first branch also, we can say that my sequence is of same species. When you introduce different species, they form different branching pattern. Okay. So once it is done, maybe PNG file or TF file, you save it because both give better resolution than JPG and PDF files. Save it, and here is your tree file sitting here. Okay. So close, if you want, you can save the tree session also. After that, minimize it and go for distance analysis, the fourth option. So once you click it, you will have a distant table. Here, before you convert them to Excel, better arrange the same species in one cluster. For example, I would say I will arrange my sequences and leuconostoc mesenterites as a first 6 sequences, like I done here. How to do it? Just click my sequence or S1 sample, whatever you have given, just drag it. Okay, you can move them along the column. Just click it by holding the sequence. By selecting it, just drag it. Okay, likewise, we can drag and you can form sequence. See, I have placed first 6 sequences of leuconostoc in one area, then gelidium together, gassicomi, palme, and lactis. I have arranged like this. Why so? Because you guys know now to click Excel and you will get a sequence in the Excel. Now, when I do a color scale bar in Excel, you see, if I arrange the sequences close together, I can simply say whichever sequence sharing less similarity, they are actually clustering here. Okay. So whichever the distance sequence, say leuconostoc lactis, it is placed here. So using colors, and we can easily identify which are closely related and distantly related. Okay. This can be saved as a JPG file or Excel file as such. So like I said in the first day itself, phylogram is nothing but a graphical representation of pairwise distance. You see all leuconostocs are in green patch, which forms single cluster as a branch number 1 in the phylogram. Okay. So likewise, we can, the lactis is actually a distant member. I can swap the tree and make lactis go to this far end also. It is possible. So we can just with the pairwise data, we can see. So pairwise data, phylogram, and blast analysis, 3 important parameters for declaring a species name. Okay. So once it is done, you will be given, people working in bioinformatics department, they are actually given this template species report where you will write the CYML decodes, okay, and date of receiving sample when you have started processing, everything is done. This is done by a wet lab department, and this is done by reception, sorry. This is done from the reception. They will decode it and they will give it to us. Wet lab department, after, once client is submitting a bacterial sample requesting for 6ins, wet lab is the one who actually select the primers and they will go for sequencing, amplification and extraction, amplification and sequencing. They will prepare this file. What is this sequence and everything about. And finally, bioinformatics department, after combining and doing the analysis, you have to put the sequence here. You have to be very precise in saying what species it is to the client. Your sample S1 is identified as leuconostoc mesenterites. That's how the email should start. Okay. So once you are done, these are the files you have to send it for email. Okay. The first important file is your notepad file containing forward sequence, reverse sequence, and merged sequence. Second important file is a species report, this CYML PDF file. So the third important file is your tree file. It is in JPG format. And the fourth important file is your pairwise distance data file. In this file, if you want, you can paste the caption of the tree you have collected before and send it to the customer. Okay. These are the 4 important files that needs to be emailed to the customers. Okay, so this is how a species identification report is being prepared. I hope you have learned something today. In forthcoming videos, we will talk about how to select a sequence for our mini project, which is I will do it today or tomorrow. This video will be circulated. Thank you.